Alright, so let's start chapter 15, which I really just say is an extension of chapter 14. But to start off, we're going to talk about these lovely pictures. But really, they lead into some different disorders. So we have this one of Prader-Willi syndrome. And so you can see, which is going with the pictures, here's little Victor at age 1, little Victor at age 2. And so he's born floppy and pale, wouldn't nurse, and then became really obese, underdeveloped sex organs, and eventually had proficiency with jigsaw puzzles. And looking at that one compared to a disorder called Angelman syndrome. So taunt, not floppy, thin, hyperactive, insomniac, small head, very happy, and severely mentally retarded. So it's rarer than the Prader-Willi. But what's interesting, so almost opposite symptoms, they're missing the same piece of chromosome 15. So what's the difference? In Prader-Willi, that piece came from his father. In Angelman, that piece came from the mother. How does your gene know this? How can it get this? So it's all coming from genomic imprinting. Let's talk about chapter 15. So in chapter 15, we talk about Mendelian inheritance. How several researchers proposed that the genes were located on chromosomes. Okay, got it. Behavior of the chromosomes during meiosis was said to account for laws of segregation and independent assortment. Sounds good. So here's a little recap on meiosis. We have our parent cells, and they go out and they get fertilized, and they go through meiosis, and we get these different gametes. And so we have law of segregation. The alleles are all separating, law of independent assortment, the different genes are separating. Sounds good. So, you have recombinations of unlinked genes, which is independent assortment. So Mendel looked at two different traits, in this case seed color and whether the seed was round or wrinkled. And so he found that there would be a combination of traits that did not match either parent. And then Thomas Hunt Morgan came along, did the wonderful fly lab that you guys have all been enjoying, and he observed that there is a wild type fruit fly phenotype. The alternative alleles were the mutants. So he was able to provide convincing evidence that the chromosomes were the location of Mendel's heritable factors. And so then we looked, and we could see we could cross these. So we had our female with, we had the females with red eyes, males with white eyes. So F1, they all had red eyes. F2 showed the 3-to-1 white eye ratio, but only the males had white eyes. So this is when Morgan proposed that the white eye mutation was carried on the X chromosome. He did several other things to look at linkage. He discovered that genes that were close together on the same chromosome are linked and do not assort independently. Unlinked genes are either on separate chromosomes or far apart, and they assort independently. Their frequency of crossing over is related to the distancing, which makes sense. So if I had a gene that was here, so if I had a gene up here and a gene up here, crossover, these two are more likely to be crossing over than a gene that's here and here, because I'm probably not going to cross over right in the middle. And so, going back, and linked genes have recombination frequencies less than 50. So, we can do a genetic map on this. So we've done a little bit of this, but many fruit flies, we use fruit flies for a lot of things. So we can look and see, based on the amount of crossing over, where we think each of the different genes are. And so this shows you that male and female with gray body, normal wings, and black body with vestigial wings. If we mix these together, this is what they believe is happening. So we have some crossing over here. And look at the different gametes and the different possibilities. And so this is the amount of offspring, how many recombinants there were, gives you the 17%. So you can use this recombinant, and this is what we did with the Sardarias, by the way. So you cross true breeding parents, and then you cross those F1 generations to see what you get. You count the recombinants, and so you can also get the sex determination. So your sex chromosomes are going to be X or Y. So in the sperm, you can either be given the X or the Y, and females can only give the X's. And so as these mix, you can see in here, this is showing how many chromosomes there are. But there are different sex determination found. So there you have the XO, so there's no Y chromosome, there's just XX, and X nothing. ZW, so there's ZZ for the male, ZW for the female, or for little B here, diploid for female, haploid for male. And then with the sex chromosomes, many genes for characters are unrelated to the sex. The genes located on either sex chromosome. So if it's on either the sex chromosome, it's called the sex-linked gene. And this goes back to our chapter, those recessive genes carried on the X chromosome. And here's some examples. So Y-linked traits show up only in males. X-linked recessive traits show up more frequently in males because they don't have a backup. Males only have one X, whereas females have two. Females would have to have both of those recessive traits in order to get it. So for a female to get, let's say, hemophilia, they'd have to have both of the recessive. So the chance of happening that is going to be a lot more rare than the males getting that one mutated gene. And the Y doesn't carry anything. So females can also be carriers. So if you think about it, they're the only ones could have. Carriers would have to be a heterozygous. Males can't, because again, they only carry one. Next, chromosomes in males flies without a co-pilot. Hee hee. Alright, alteration. The breakage of the chromosome can lead to 4 types of changes. So if something goes wrong, you can get deletion, duplication, inversion, or translocation. So in here, we have a duplication. So here's the gene. Oops, went in. Now I have more because of so in deletion, I deleted it out. Duplication, I accidentally copied it twice. Makes sense. So a deletion is going to occur when a chromosome fragment is like in the centromere. So a chromosome will be missing certain genes, whereas the duplication occurs when the fragment is attached as an extra segment to that sister chromatid. It can also have inversions, which is where I flip-flop things. So you can see the flip-flop. And translocation, the fragment joins the known homologous. So like if we're thinking X and Y or something, or a completely different chromosome, these rotated. So the translocations can happen between non-homologous. So like here show the difference of chromosome 22 and chromosome 9. So that can happen in this type of leukemia. And you can see that here. So this is way too long. And so you can see similar banding patterns till you get right there. So this again is the karyotype. And this is actually one with Down syndrome, but instead of having a third 21, or that 21 came up here. So pretty interesting. There are different ones that have these. So Cry to Chat, which the kid actually sounds like they're crying like a cat, is from a deletion in chromosome 5. So the frequency of aneuploid, so less zygotes, could be quite high. And so, but most of these are going to be aborted, not by a person, but your body. So miscarriage long before birth, and so it upsets the balance, and these kids can go to birth and beyond. So they have a set of symptoms characteristic of aneuploidy. And non-disjunction. So this one is the failure of a homologous chromosome to separate. And so we get these guys who are all supposed up here, they were supposed to separate, they didn't, and so this just went in half, and we get trisomy. Over here, they were supposed to separate, so you're going to get trisomy. You're going to have 3 once mom comes in, or monosomy. So, non-disjunction is results from the chromosomal aberration, where you have extra or missing copies. So that's the aneuploidy, when you have extra, like over here I have extra. So a cell with only one, mono meaning 1, trisomy could have 3. So going over here, so you can see here I end up getting 3 and 1, and so now I could have trisomy. There's several different ones, so Down syndrome and extra chromosome 21, Klinefelter's, XXY, Turner, 1X, XXX, and XYY in males. So here's Down syndrome with your extra 21. And some characteristics about Down syndrome. Can happen in 1 in 800 births. And you can read the different syndromes. Turner syndrome, they only have 1 X chromosome. And so she's female, but usually small, hearing loss, and don't usually develop. So she wouldn't be able to have kids. XXY, and so 2 X's but a Y. So it is male. And they're usually taller. They have male sex organs, but they are sterile. Again, can't have kids, and they could have some feminine characteristics. And usually don't discover it till puberty. XXX, and so 1 in 1,000. Normal IQ, but might be a little lower than their siblings. They do have some learning disabilities, but mostly clumsy and a little bit more introverted. XYY, usually taller, normal, and maybe a little bit more learning disabilities, but really not too much effects. So, organisms that have more than 2 complete sets of chromosomes have gone polyploidy. So, organisms that no normal gamete fertilizes another, there's been non-disjunction in all the chromosomes. So it'd have to be a triploid. You'd actually have 3N. And so if that 2N failed to divide at all, you could have a tetraploid embryo. A polyploid is much more common around plants, but you can get a normal phenotype, and then one extra or missing chromosome usually upsets the genetic balance during development more than the entire extra set. It's kind of interesting. We're going to have to stop now, but we just have a little bit more for the next PowerPoint.